Welcome, everybody, and thank you for joining us for today's session on pioneering our understanding of the human brain. Throughout the day today, we will be privy to the most up-to-date research, issues, and innovations in our quest to understand human brain function. This session is par excellence due to the participation of Brain Initiative. We begin today with the scientific updates track, which will bring us au courant with the latest progress in the research on humans division of brain. The Innovative Neurotechnologies track will showcase supreme cutting-edge innovations which will foster a dynamic understanding of brain function and provide new hope to patients. We'll have important discussions and hear from top experts on the ethics of using such technologies and the impacts of our advancements to ensure that these objectives continue with best practices in the neuroethics track. Finally, we'll round out the day by hearing from clinician scientists on how the application of the latest neurotechnologies benefits patients now, while laying the groundwork for improved healthcare and personalized medicine in the future in clinical updates. And now, it is an honor and a pleasure to introduce the person who will launch today's momentous session, Dr. Joshua Gordon. Dr. Gordon is the director of the National Institute of Mental Health. He received an MD-PhD degree at the University of California, San Francisco, and completed his psychiatry residency and research fellowship at Columbia University. He joined the Columbia faculty in 2004 and remained there until 2016, when he took over as director of the NIH. During that time, he also had a private practice, was an attending at the New York Presbyterian Hospital, and was the director of neuroscience education at New York State Psychiatric Institute. Dr. Gordon's work has been recognized by several prestigious awards, including the Brain and Behavior Research Foundation NARSAD Young Investigator Award, the Rising Star Award from the International Mental Health Research Organization, the A.E. Bennett Research Award from the Society of Biological Psychiatry, and the Daniel E. Efron Research Award from the American College of Neuropsychopharmacology. For a complete biography on Dr. Gordon, please visit the Biography tab at the top of your screen. Dr. Gordon's talk today is entitled Updates from the NIH Brain Initiative. My name is Dr. Roshanak Hashemian, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would like to extend our special thanks to the Brain Initiative Program Directors, and in particular, Dr. James Knapp, for their efforts in organizing today's session. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting questions during the presentation. Simply click on the Ask a Question box located at the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions following his presentation. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. And now, without any further ado, please join me in welcoming our keynote speaker, Dr. Joshua Gordon.
Hi, thank you, Dr. Hashemian. I appreciate the introduction and appreciate the opportunity to talk to you all about the NIH Brain Initiative. I'm really here on behalf of a whole number of staff at the National Institutes of Health and indeed across government that has been working diligently on the Brain Initiative since its inception about 6 years ago. The Brain Initiative organization is run nominally by myself and Dr. Walter Koroshetz, who is the director of the National Institute of Neurologic Disease and Stroke, and we serve as co-chairs of a board of NIH Institute directors. But of course, the Brain Initiative itself is bigger than just the NIH. I'll be focusing mostly on the NIH portion, but we'll discuss interactions with other government agencies and indeed international brain initiatives towards the end of my talk. What is the Brain Initiative? The Brain Initiative is an attempt to see the circuits of the brain in action, to try to understand how those circuits control how we move, plan, execute, remember, emote, and otherwise perform all the functions that we do as human beings. But in order to see those circuits in action and understand how they control our behavior, we need to develop novel technologies. We need to develop these technologies that will help us monitor and manipulate circuits, monitor them to figure out how they work, and manipulate them to improve function in case of disease. We also need to develop these technologies in order to understand how disordered brain circuits cause neurological, psychiatric, and substance use disorders. The BRAIN Initiative was started, as I said, about 6 years ago by the President Obama at the time, and it was immediately taken up by many agencies within the U.S. government and indeed private entities as well. The first thing that had to happen when the BRAIN Initiative started was we needed to develop a plan. So in 2013, President Obama's Office of Science and Technology Policy created a work group with the help of the NIH director, Dr. Francis Collins, and his advisory council. That work group set out developing a scientific vision. That vision essentially created a plan that was divided into the first 5 years of the BRAIN Initiative, as delineated by the vision, which was titled BRAIN 2025. The first 5 years were meant to emphasize technology development. The second 5 years were meant to emphasize discovery-driven science using those technologies. We've actually done a fairly decent job of getting things started. I'll tell you about that today. And then I'll talk about how we're strategizing the pivot from technology development to discovery-driven science as we approach the real halfway point of the 10-year period from 2015 to 2025. Of course, the BRAIN Initiative would have amounted to not much at all were it not for the commitment on the part of Congress to fund the initiative. The initiative started out with modest funding, but over the past 5 years, that funding has increased to the point where in this fiscal year 2019, we have about $430 million a year to spend, and that budget has been committed through 2026. That $430 million, did I say 3,000? $430 million to spend. That budget has been committed, as I said, through 2026. And also importantly, it meets actually the recommendations for the budget for the BRAIN Initiative set forth in the 2025 document. Those funds are meant to be distributed across a range of different areas, everything from discovering the various cell types of the brain, from developing tools to interrogate those cells and the circuits they compose, to developing novel tools and methods in human imaging and modulation of neural activity in human beings, as well as more granular methods to look at neural recording and modulation in model organisms. Tools and processes to use them in order to develop a better understanding of the neural circuits themselves. And then 3 overarching components of the BRAIN Initiative: a training and dissemination component. We recognize that we need to be able to train the next generation of scientists to continue developing these tools and even more importantly, to use them. We need to disseminate these technologies so that they are broadly available in the neuroscience community. Then a second overarching area of data coordination and informatics, which we're really just getting started. We intend that all the data gathered through the BRAIN Initiative be shared. And in fact, we have been given authority by Congress to require such sharing. And as of next spring, any submitted BRAIN grant will have to promise to share all the data from that grant, as well as the software used to analyze that data through the networks that we're constructing in the BRAIN Initiative. And the third overarching area is neuroethics. We recognize that many of these technologies, when applied to human beings or even to animals for that matter, raise novel ethical issues that need to be addressed, particularly as these technologies move into the dissemination phase. So that's the overview of the budget and what we're spending it on. As I said, the budget has been committed through 2026, and you can see this in this next slide. The total committed by Congress to the BRAIN Initiative is about $4.9 billion, which represents, as I said, something close to what was recommended by the 2025 vision, which was $400 million to $500 million a year for 10 years. We have given out, importantly, much less than half of that total in the first 5 years of the BRAIN Initiative from 2014 to 2018, which means the best is yet to come. And there's lots of opportunities to continue the rapid development of technology that we've initiated, and importantly, to disseminate that technology and implement it throughout the neuroscience community. We recognize, of course, that as we approach 2020, which is the nominal halfway point of the initiative, that we need to examine the progress so far and determine what the best ways forward are to build on that progress. And while the scientific vision delineated in BRAIN 2025 still guides us, and we imagine will continue to guide us through 2025 or 2026 when that funding ends, but we also recognize that we need to reconsider it. And so about a year ago, Dr. Collins, the NIH director, reconvened a new working group to take a look at the goals of the scientific vision delineated in 2013 and figure out how we need to tweak them, what goals can be expanded because we've made more aggressive progress than we anticipated, and what goals we haven't made so much progress on, so that we need to reconsider our approaches. And then finally, what new things can we imagine doing now that we couldn't have imagined 5 years ago that we should be thinking about as we push towards 2025. And I'll have more to say about that work group in a few moments. These are, again, the 7 areas of focus of the BRAIN Initiative. The first, tools for cells and circuits, well, first let me say, and in each one, we have a number of funding opportunity announcements that are open for individuals to submit grants to. And of course, in each one, we also have active grant programs. And I'll show you some of the advances that many of these programs have resulted in, just to give you a flavor for what we've been able to do with the BRAIN Initiative funds at the level of the NIH. I should mention that although this strategic vision serves to guide more than the NIH, I'm really going to focus on the products and progress that have resulted from NIH grants. There are important contributions to the BRAIN Initiative made by agencies like DARPA, the Defense Advanced Research Technology Administration, as well as the National Science Foundation. And I'll have more to say on those partners and others towards the end of the talk. So the first area, the cell diversity and tools for cells and circuits focus, has 5 funding opportunity announcements up. And really these focus on technologies that will allow us to understand what are the building blocks of the brain and where are those building blocks. So for example, we've launched a major initiative called the BRAIN Initiative Cell Census Network, which seeks to catalog all the types of cells there are in the brain, both neural cells and non-neural cells, and figure out the diversity not only brain-wide, but also region by region. And it's really tasked with building a 3-dimensional atlas of the brain. We have a number of funding opportunity announcements in advanced technologies for neural recording modulation and stimulation. And this really focuses primarily on model organisms and on human beings. In model organisms, Brain Initiative investigators have constructed technologies that allow us to monitor thousands, tens of thousands of neurons at the same time, whether it be electrically or optically. They've developed technologies to be able to modulate specific neurons at specific moments in time and in specific spatiotemporal patterns. And novel technologies that have the potential to be used non-invasively in humans to give you some of that same cellular specificity, like ultrasound coupled with mediators that might be expressed in cell-specific patterns. On the neuroimaging front, which is a major component of this, advanced technologies in neuroimaging are giving us greater specificity. And in the brain stimulation front, as I'll show it later on in the talk, Brain Initiative investigators are developing closed-loop stimulation paradigms, for example, that allow greater control over symptoms such as tremor in patients and Parkinson's disease and in benign essential tremor that really hold out the promise to bring a new age of deep brain stimulation to bear on clinical problems. Each of the other areas has a number of active funding opportunity announcements. I'll just point out a couple more. In the understanding neural circuits area, we're trying to encourage collaborations between labs to use the technologies that are developed in the previous two areas to fully delineate the structure and function of individual neural circuits that are responsible for generating specific behaviors. We're really open in terms of the circuits that are being used, and one area that we really hope to see some progress in are the circuits that control the expression of pain. This is obviously an area of great clinical need, and it's an area that's often neglected in neuroscience, and we don't really have a good handle on, particularly on the central circuits involved in pain control. And so we're hoping to make some progress there, and we have a specific announcement out to ensure that people understand that just because we are the Brain Initiative that's focused on technology development and understanding neural circuits, it doesn't mean that you can't exploit them to study circuits of clinical relevance. I should also mention one more that I didn't mention before, which is that we have a funding opportunity announcement at the bottom of the page for small businesses to use brain funds to further develop these technologies and commercialize them as one pathway towards dissemination, although there are certainly others that we are supporting. I mentioned the Brain Initiative Cell Census Network. Again, the goal of that network is to provide a comprehensive reference atlas of all the different cell types in the human, monkey, and mouse brain, and to understand that brain by brain, but also region by region and area by area, and eventually to understand how those cell types assemble into specific circuits. We've invested in a number of different projects that total about $250 million to be invested over the next 5 years. These funded projects will support a brain cell data center that will aggregate the data from all the projects, make them publicly available in a usable interface, a mouse brain cell census center that focuses on establishing all the brain types for the mouse brain, as well as a brain cell census collaboratory, an opportunity for the various investigators who are carrying out these projects to work together collaboratively to match their results across labs. And then a similar collaboratory for human and non-human primate brain cell censuses, which for now anyway, are focused on specific parts of those organisms' brains, but will eventually hopefully achieve the full brain scale that we are currently achieving with the mouse brain cell census. The anticipated outcomes of this Brain Initiative cell census are the essential knowledge of the diverse cell types of the brain, and really crucially, their 3D organization within the brain. The goal of the Cell Census Network is to create an open access 3D digital mouse brain cell reference atlas that you'll be able to poke and prod and find out where are the different cell types, which brain regions, where do they project to, etc. That will yield a comprehensive neural circuit diagram for the mouse brain. The eventual goal that we hope to achieve through the BRAIN initiative is to create the same kind of atlas for the human brain and for at least 2 non-human primate species. But first, we need to figure out how to do this on a smaller scale, and the mouse is the model for that. This atlas will be tremendously useful for neuroscientists studying the mouse, and indeed studying other organisms as well. To have established a complete circuit diagram with all the component parts will be kind of like having the human genome sequenced. We will know the players. We'll know the size of the field. The details will be important. We'll need to really truly understand not just what cell types there are, but how they change with activity, how they change through development, etc. But the first start of characterizing an atlas of all the components of the brain is an important one, and that's the main goal of the BICCN. What does it take to build such an atlas? It takes an understanding of the shapes of the brain, the morphometry of it. It requires an ability to position compartments in relation to the various components of not just the neural structures of the brain, but the neurovascular units. It requires knowledge of the anatomical projections of dendrites and axons in both inputs and outputs of brain regions. It does require some understanding of developmental migration, particularly if we are to be able to integrate an atlas with development, with what we know about development, and eventually create developmental atlases as well. But it also requires an understanding of the cell properties themselves, the molecular properties, their transcriptomes, their proteomes, but also the physiological properties of those cells, passive membrane properties, the complements of ion channels and neurotransmitters they use, and the receptors that they have. And then finally, we don't just want an anatomical atlas, we want an atlas that we can build over time a functional component to, that is, what the role of those circuit components are in activity and how they change based upon the history of the organism and the history of the brain. So what we're creating is a structure that initially will be cell type and anatomical and some physiology, but that we can add to and build to over time so that eventually it's really a truly comprehensive encyclopedia of the knowledge that we have about the components of the brain and how they generate and are related to behavior, and how they change over time. What's our vision for the time frame for this cell census? Well, the first grants are actually complete. They were pilot grants that were aimed at developing the technologies to be able to create this cell census, this cell atlas. And that was completed in 2016. And what surprised us was the rapidity with which novel technologies were developed, particularly single cell technologies such as DropSeq for transcriptomics, but also single cell epigenetics, and other important advances like high throughput cell physiology and high throughput anatomy. These advances were made so quickly that we were able to begin the mouse cell census project earlier than we had anticipated. And those grants, as I said, have already been given out with $250 million to be spent over 5 fiscal years, from fiscal year 17 to fiscal year 21. And we hope to have at the end of that a complete mouse census. And while that is happening, we will be ramping up, as shown in the red line, funding for human and non-human primate cell census efforts as the technologies mature and as we gain the knowledge from the mouse projects about how to proceed with these larger projects. So the BRAIN Initiative Cell Census Network is really a flagship product of the BRAIN Initiative that has 2 important aspects to it that I want to emphasize. First, it demonstrates how a concerted focus on technology early in the course of an effort to understand the brain can yield tremendous benefits. We were able to ramp up the mouse cell census project much, much more rapidly than we had anticipated because the technology developed so quickly. And so that enabled us to take advantage of these changes of technology and achieve what we think is a tremendously ambitious set of goals. The other aspect of the project that is important to emphasize that represents what we're trying to do really Brain Initiative-wide is create technologies and resources that the entire community can use. So at the conclusion of the mouse cell census project, there will be an open source atlas of cell types for the mouse brain. How much information it has, that depends upon the success of the project, but it will be available for all investigators to use, and it will really enrich neuroscience broadly. So creating these tools and technologies and doing it ambitiously and with a focus initially on technology and a second focus on exploiting them. This is the success that we hope to replicate in other areas of the Brain Initiative. And that's why I wanted to focus on the Brain Initiative Cell Census Network for a time. There's lots of other investigators in the Brain Initiative, funded by the Brain Initiative, who have done really amazing things. And one area where there's also been tremendous advancement is in the area of cell and circuit tools that are aimed, as opposed to defining cell types, aimed at understanding their function. One example of that is illustrated here. It's a microscope, or actually a dual microscope, that allows simultaneous wide field imaging and narrow field imaging in mice and in lower organisms that allow one to capture both broad activity throughout in the case of the mouse, the surface of the brain, as well as narrow activity within a specific brain region, to look at the relationships between activity patterns of single neurons with activity at scale across the entire brain. And these are the kinds of technologies that will allow us to really understand the coordination of neural activity across broad swaths of brain and understand how circuits integrate into larger neural systems. These images, I wanted to show you some pretty videos from them, but I guess they didn't carry over, so we'll skip over to the next one. I mentioned, of course, that there is a big focus from the Brain Initiative on human neuroimaging and on developing novel techniques to modulate activity within the human brain. And although our focus is on basic science and although our focus is on technology development, this human component is important because as quickly as we can, we want to try to allow for the translation of these technologies into clinically relevant projects. So the Brain Initiative has supported next generation human imaging technologies that promise to give us better and faster pictures of human brain activity. We need to understand the actual foundations of activity that is determined by current methods for human imaging so that we can improve upon those methods and perhaps more importantly, develop novel methods of capturing that activity. So we've funded studies that seek to understand the relationship between neural activity measured electrically at the level of single neurons and at the level of local field potentials with the activity that is simultaneously achieved with non-invasive methods. We also seek to improve our ability to conduct non-invasive neuromodulation, to look beyond the current technologies at new technologies that might be able to modulate neural activity. I mentioned ultrasound before, that we have a significant investment with several groups carrying out experiments to try to refine ultrasound-based neuromodulatory technologies. And an important advance that's already making clinical progress, we're supporting the generation of the development of the next generation of deep brain stimulation devices for a range of disorders, including pain, including depression, and including control of benign essential tremor. And there, in a really remarkable series of experiments, Brain Initiative and funded investigators have developed a device that can monitor neural activity in the motor system to determine when an individual is making a movement. In benign essential tremor, the tremor is not present at rest. It's only present when an individual is making an intentional movement. And these devices can record activity, determine with an algorithm when that individual is about to make a movement, and then turn on the stimulation that reduces the tremor when and only when the individual is going to have that tremor. That is when that individual is going to make an intentional movement. These devices have already been implanted in a handful of patients and really are working quite well. And this is important for several reasons. Number one, that the constant stimulation that's present in these devices does cause side effects. So obviously, if you only have the stimulation running when you need it, then those side effects are reduced. It also improves the battery life of the device. And of course, changing out the battery requires a surgical procedure, a minor one, but a surgical procedure nonetheless. And it also serves these record and stimulate devices, these closed loop devices serve it as an early example of the potential successes that one gets when one uses the information that one can gain from recording from the brain to help customize clinical treatment for patients. And as I said before, studies are underway in depression and in Parkinson's, supported by the BRAIN Initiative, both at the NIH and other federal partners, to try to use closed loop stimulation and or precision medicine determined by recordings from these deep brain electrodes to design brain stimulation parameters that will help the patient better and make efficient use of the technology. Not surprisingly, the investigators supported by the BRAIN Initiative have been making such wonderful discoveries that they've been recognized by a number of awards and accolades. Most recently, the National Academy of Sciences recognized 3 of our brain investigators. Li-Chin Lo of Stanford won the Piradell Research Award. Xiaowei Zhuang of Harvard won the National Academy of Sciences Award for Scientific Discovery, and Eve Marder of Brandeis University won the National Academy of Sciences Award in the Neurosciences. These awards were given for the expertise that makes these individuals great brain investigators. So Li-Chin Lo for addressing how serotonin modulates diverse physiological functions and behaviors using viral and genetic tools. Xiaowei Zhuang for the efforts in super resolution imaging and single molecular imaging and genome scale imaging that are helping us create that spatial atlas, that cell census and cell atlas of the mouse brain, and Eve Marder for understanding flexibility, stability, and neuronal microcircuits. And Eve is a member of the multi-council working group for the BRAIN Initiative. That's the group that has been advising the institute directors and Dr. Koroshetz and I on the grants that we give out and the funding opportunity announcements that we provide to the community. The BRAIN Initiative is not just about the technologies and the science. The BRAIN Initiative is also about developing the next generation of scientists. And in some ways, the BRAIN Initiative has been tremendously successful at the level of diversity, of ensuring that our scientific workforce has diversity, but only in some limited areas. And particularly those areas are where we've been successful are in bringing engineers, computational scientists, and mathematicians and others into the neuroscience sphere, bringing their expertise to bear on neuroscience questions. And we admittedly have been less successful at ensuring the ethnic and gender diversity and racial diversity of our investigators. And so we've begun a series of outreach efforts to try to enhance the diversity of investigators interested in these cutting edge projects in neuroscience. Led by Dr. Michelle Jones, the National Institute of Neurologic Disease and Stroke. We've begun efforts to reach out to minority students to encourage them to participate. These include presentations at the annual Biomedical Research Conference for minority students and upcoming presentations at various societies, including the Society for Women's Engineers, Society for Advancement of Chicanos and Hispanics and Native Americans in Science, and other societies to try to inform and encourage these students to take advantage of the opportunities the BRAIN Initiative offers. As I mentioned at the beginning of the talk, we recognize that as we approach the halfway point in the BRAIN 2025, we need to examine what we've done and think about how we can improve upon that, and also need to really begin planning for this second 5 years, which is meant to emphasize discovery-driven science as opposed to technology development. So Dr. Collins recruited a new advisory group to look at the BRAIN 2025 report and try to figure out what we've learned so far that we can build upon more aggressively, where we haven't made such great progress, and where there are new opportunities given the technologies that we should investigate beyond what was envisioned 5 years ago. This effort has been led by two outstanding neuroscientists who've been involved in the BRAIN Initiative for a long time, Dr. John Munsell and Dr. Catherine Dulac. The group convened just under a year ago at the BRAIN PI meeting. That's the meeting of the BRAIN principal investigators, which occurs once a year, when we celebrated the 5th anniversary of the launch of the initiative, and announced the creation of this working group. And this work group is actively deliberating how to revise the 2025 report, and also with a subgroup of that work group to examine how to expand the neuroethics efforts and make sure that these neuroethics efforts and investments are solidly covering the various areas of the BRAIN Initiative. The overall work group had an aggressive timeline. As I said, it was convened in April of 2018 with a public town hall at that BRAIN principal investigators meeting. Shortly afterwards, the work group began having a series of teleconferences amongst themselves and planning the outreach efforts that they would do in the summer. The NIH put out a request for information to the neuroscience community, asking for input on BRAIN 2025, on what new things should be included, on what we've been successful about, and what areas we might go into. Over the course of the summer of last year and the fall of last year, we began a series of country-wide workshops. There were, I think they're here. Sorry, I had a problem with my screen here. There it goes. In August of September and October, in Boston, in Chicago, and in Houston. And each of those workshops focused on a different aspect of the BRAIN Initiative. Then in the fall of 2008 at the Society for Neuroscience meeting, we had a second town hall, a larger one, attended by literally, hundreds of people at the Society for Neuroscience, who had the opportunity to give input. And in December, a preliminary report, an interim update of the work group, was given to the Advisory Council for the Director with a series of high-level review items. Based on that feedback, the committee is now engaged in writing a draft of their recommendations, which will be released for public comment. And the plan for that will be to finalize a revised strategic plan that will be submitted this summer in anticipation of being able to prepare funding opportunity announcements that are responsive to that strategic plan for the next fiscal year for 2020 and beyond. So that's the plan of the work group, and they are hard at work, and we're really pleased with the progress they've been making. As I mentioned, as part and parcel of that work group, a section of it is focused on neuroethics. In the meantime, though, we've not been idle. The BRAIN Initiative has had the multi-council working group, which is the advisory council for the BRAIN Initiative, has had a neuroethics subgroup as well, and they've helped us issue a series of guiding principles for neuroethics. This has been published in the literature, in the Journal of Neuroscience, detailing the focus on safety in terms of developing these technologies for human beings, ensuring that these technologies are monitored and that ethical issues are dealt with, particularly those that relate to capacity, autonomy, and agency, that we protect and provide provide confidentiality for neural data that is shared through the BRAIN Initiative networks, through the BRAIN Initiative databases. that we think about the potential malign uses of the neuroscience tools and neurotechnologies to guard against them. That we move tools and technologies from research into medical, that caution should be taken. We at the NIH focus on using these tools and technologies to improve function in disease, and that we believe that should be the focus for the BRAIN Initiative moving forward, and that any moving of use of these tools and technologies into non-medical uses should be with caution, and into medical uses should be with an eye towards ensuring equity and other ethical principles, as I mentioned before, capacity, autonomy, and agency. We also need to ensure that we continue to identify and address the concerns of the public about the BRAIN Initiative and BRAIN technologies as they arise. And that means encouraging public education and dialogue, and that we should ensure that the technologies that are developed and their clinical applications that are developed through the BRAIN Initiative, that they are shared justly and equitably. Of course, neuroethics is not just an issue for the US BRAIN Initiative, it's really a global issue. And so we have participated in an annual Global Neuroethics Summit that was convened first in 2017 and then again in the fall of 2018, with an eye to ensure harmonization of neuroethical principles across the various brain and neuroscience initiatives that are taking around the world. We've also been collaborating with those various BRAIN initiatives in the form of a group called the International BRAIN Initiative that was convened by the Kavli Institute. It's been a big supporter of the BRAIN Initiative since its inception, or the Kavli Foundation, I should say. There are two main efforts that we've been involved with to date, which the first is that neuroethics effort, and the second is an inventory of global brain projects that has been carrying out under the auspices of the Kavli Foundation to try to understand and describe what the various national brain initiatives and brain projects are focused on as a stepping stone to building out other aspects of the International Brain Initiative, including assisting international dissemination in tools and technologies, development of international data standards, and ensuring data sharing across international boundaries, communication and outreach, and education and training. And each of those areas is currently in what we call the scoping phase. We're exploring them with our colleagues around the world. Finally, as I approach the close of my presentation, I wanted to make sure that everyone listening in is aware of next month's Brain Principal Investigators meeting taking place April 11th through 13th at the Marriott Wardman Park here in Washington, D.C. If you're interested in gaining information and registering, please, you can click the link here or copy it down. We have some really exciting keynote speakers, both Brain Initiative investigators and others, and it's an international group. So we have Paula Arlotta from Harvard University, who works on organoids and other iPSC line-derived technologies, but also participates in the cell census network projects. Edvard Moser from the Kavli Institute for Neuroscience, who of course is a Nobel Prize winner and focuses on hippocampal circuits and entorhinal circuits and their role in encoding spatial information. Patricia Churchland of the University of California, who is a well-known philosopher of neuroscience and has dealt with many of the thornier issues, such as agency that are brought up by the neuroethical issues in technology, and Marcus Meister from Caltech, who is a pioneer in developing novel technologies to monitor and manipulate brain activity. So it's really been quite an adventure participating in this BRAIN Initiative. Our efforts to map brain activity at the speed of thought to ensure that we have the best technologies is really going full speed, and I'm happy to answer any questions that you might have. Thank you.
Thank you, Dr. Gordon, for that outstanding presentation. We will now begin the live Q&A portion of this presentation. As a reminder, please submit your questions for Dr. Gordon by clicking on the Ask a Question box on the far left of your screen. We will address as many questions as time allots for. So we do have a few questions here that I'd like to share with you, Dr. Gordon. The first came from, let's see, it says that how much support do you anticipate grants allocating for non-human models, specifically zebrafish, which are extremely cost and time effective for experiments very similar to rodent capabilities?
That's a very good question. I emphasized in much of the work that I showed the mammalian aspects of the BRAIN initiative, but we have a number of projects that support, for example, whole brain imaging in zebrafish and in flies, particularly those that are focused on simultaneous imaging of entire circuits and behaviors. The question asked about funding levels, and it's important to recognize that we don't have preconceived notions about how much money should be spent on any one organism or any one circuit. We look for scientific opportunities as they arise. The goals of the BRAIN initiative are to enable the eventual monitoring and manipulation of circuits in the human being, because we in neuroscience are generally focused on trying to understand how it is that we think and produce behavior. But we recognize that a range of different models are necessary to answer the questions that will eventually get us that knowledge about how and why we think. And zebrafish and other non-mammalian organisms with complex brains, complex enough to be able to ask really crucial questions about how circuits operate, they play a very, very important role. So we have funded a number of projects in these organisms.
We have another question, and a second question from another attendee that's related. So I will ask you both of them together.
Sure.
What are the opportunities within the BRAIN Initiative for early career researchers who are not citizens of the U.S.? And the second question is, will the BRAIN Initiative be open to beyond U.S. collaborations? What will be the impact of models and deliverables and their counterparts from the EU's Human Brain Project?
Those are two great questions. I'm not 100% sure for the first individual about the non-citizen question, where it comes from. There is a BRAIN Initiative K99 program that is for underrepresented minorities, and I believe that may be restricted to U.S. citizens, but I'm not 100% sure. The general K99 program, though, is not restricted to U.S. citizens. Early stage investigators and faculty positions who are not U.S. citizens, I shouldn't say not U.S. citizens, but really you have to be a U.S. citizen or a green card holder for some of our training programs. And with the exception, as I said, of the generic K99R00 program that applies to most of our training programs. But for early stage investigators who are faculty, there is no restriction as such, and so those are completely available to individuals who are not U.S. citizens. And this gets me to the next question. The BRAIN Initiative actually has given grants to international investigators. So the BRAIN Initiative is empowered to grant funds, as are all the institutes, to investigators in other countries that are answering the questions that need to be answered, where they're the best situated to answer those questions, or in the case of the BRAIN Initiative, to develop those technologies. We do also support a number of collaborations that cross national boundaries, and some of those do involve investigators who are also engaged in Europe's Human Brain Project. So I would say there's a lot of synergy in terms of the grants that were funded. And of course, there's a lot of synergy intellectually. The European efforts focus on models of the brain, on computational models, is very, very complementary to our efforts to try to use those computational models to address circuit level functioning. So we know that there's a lot of crosstalk, and we anticipate that only increasing in the future.
Thank you for that. We have another question. The Brain Atlas sounds amazing. You said phase 1 is already complete. When do you project it will be available for us to see? And is it analogous to the 3D human brain coming out of Jülich in Germany?
Yeah, so I would say it's analogous, but it'll be, we hope, much richer in terms of the data that is included. Although that effort is a really important one that has made that is a really wonderful contribution. It's similarly analogous to the Allen Brain Atlas that exists already that describes expression of for example, of proteins and RNAs throughout the brain. And has a lot of anatomical data as well. But what we really hope to achieve is a neuron by neuron atlas. Some of the data is becoming available already or will be shared shortly. The full project will be complete in 2021, and I'm not exactly sure when you'll have a usable 3D atlas in that time frame, but the hope is by 2021 it will be available. But the data behind it are being made publicly available on a more aggressive timescale.
Thank you. We have another question. Given that you are now allowed to and even required to share all the data that comes into the research from the BRAIN Initiative, how will you be able to handle the tremendous amount of data that will arise from that research and process it in a coherent manner?
Well, it's a challenge. I'm not going to lie. We've put out really the data sharing infrastructure is something that needs to be built. So at the NIH and at my own institute, the National Institute of Mental Health, we've developed data infrastructures that can handle very large data sets, really petabyte size or hundreds of terabyte size data sets from human neuroimaging. So we have developed the capacity to share very large data sets and to store them. The BRAIN Initiative data sets that will come in, because they are activity over time, are going to be perhaps an order of magnitude larger and more challenging to share. But as I said, the grants to begin developing not just the software infrastructure, but also the protocols, the standards, and the methods, frankly, have all been awarded, or the first layer of them, I should say, have been awarded, and we hope within the next year or two to be able to stand up some of those structures and facilitate some of those protocols. I should mention, though, that one important aspect of this sharing opportunity is the fact that you're probably going to have to do your data analysis on these large data sets in the cloud. The data sets now are getting too large to download. That's already true for some of our larger human neuroimaging data sets, like the Adolescent Brain and Cognitive Development Study, where we can't download it. They're too big. You have to, if you want them brought in, you have to pay for a hard drive to be shipped to you. So with these brain data sets, we imagine even more importantly, the cloud computing is going to be the way to go.
Okay, great. I'm just going to bring up one more quick question, because I think this is extremely important given the mental health crisis that we're facing. If you don't mind, we only have a few more minutes. How have the fruits of research from the BRAIN Initiative contributed to our understanding of opioid dependence and potential ways to overcome it?
I think we're at the early days of contributions of the BRAIN Initiative in that area. As I said, we did put out a notice saying that we want to be able to use BRAIN Initiative technologies to study the neural circuits engaged in pain, and of course that will also influence our understanding of how the brain circuits are affected by opiates. The National Institute of Drug Abuse has a number of projects in that area, and they have a number of investigators who are NIDA funded, who are engaged in the BRAIN Initiative, developing the technologies to improve our ability to study it. So I would hope that we would have some answers in that realm in the near future.
Okay, great. Thank you very much for that. There was a question also if this presentation of yours will be available later on by attendee Chris. I just wanted to let him and the others know that this talk will be available on demand for the next 3 months So for anyone who came in partway through Dr. Gordon's talk, it's still available too. Thank you again, Dr. Gordon, and thank you to our wonderful audience today for your incredible questions. Any questions that were not answered will be followed up by our speaker via email. Coming up next, Dr. Jim Knapp and Dr. Karen David will be discussing NIH brain funding opportunities, specifically the NIH Brain Initiative and functional human neuroscience. Hope to see you there.